So good evening, respected faculty members, and to all the participants joining us live on this evening
You're
through
on mute.
our YouTube channel.
Hello.
Hello,
Yeah, Seema.
am I audible?
Yeah.
Okay. So good evening, respected faculty members, and to all the participants joining us this evening live through our YouTube channel. A very warm welcome to each one of you to our fifth webinar series from the team Nephrocon. Well, as we gather virtually once again, we are reminded of the incredible community that has formed over these webinars. Your active participation and engagement have contributed to the success of our previous webinars, and we are eager to continue this journey of knowledge sharing and collaboration. You all are encouraged actively to participate in discussions and share your thoughts in the YouTube live chat box. The theme for today's webinar is Omics and Transplantation, and we thank our distinguished speaker and esteemed panelists who have generously shared their time and knowledge to make this event possible. So I think without any further delay, let's get started. I would like to welcome and invite our speaker for today's webinar, Dr. Satish Balan, sir, Senior Consultant Nephrologist at KIMS Hospital, Trivandrum, and I will request sir to kindly initiate with his talk.
So good evening to all. I hope you can all hear me clearly.
Yes, sir.
Yeah. Okay, so today we are going to talk about omics and transplantation. I think it's a very important topic because genetics and omics are becoming more and more important. We are, even about 5 years ago, I think we were not really into omics. Genetic tests were very expensive. Hardly we used to do one or two in a year or so. But over a period of time, these tests have become cheaper, access has become better, more and more labs are cropping up with very good validated genetic platforms. So it is now possible for us to do a lot of omics testing, which is not only is feasible, but is also practical. And the second point is that these technologies are changing transplant as we feel. Because there is a lot of advances in transplant. Even if you look at that part of the thing, even a few years back, we were not doing extensive immunological evaluation in the transplant recipients. We would not be doing, say, single antigen bead and all that. Nowadays, we carefully do single antigen beads in selected patients. So these things are all advancing, and omics is another feather in our cap. Probably we should be utilizing omics technologies more and more. So there are a lot of omics technologies which are present now and which are going to be there in the future, and I will be dealing with all of them. So basically, I've structured this as the journey of a fictitious case, because there are certain things which happen to this patient which are still not available for us, but I have put it as part of this patient's progress over time. So that will be the thing. So first of all, what are omics technologies? So omics technologies refers to the entirety of one particular platform that you're going to see. For instance, genomics means you are going to evaluate the entire genome or part of the genome. And secondly, you are going to not only identify a genetic variant by DNA sequencing, but also you are to do coding, non-coding regions, and identify all the other genetic abnormalities present in that patient, in that genome. Then there is epigenomics, which refers to the epigenome, that is the DNA methylation, the histone content, all these things you can evaluate as part of the epigenome. Then if you are measuring the mRNA especially, that would be called as transcriptomics. So because it sees what is the transcriptome. The transcript is actually the mRNA, which is transferring the message of DNA to the protein or the protein synthesis. So transcriptomics measures actually, if you look at the mRNA, it means the active DNA in that, because only the active DNA is producing mRNA. So the DNA which are actually being actively utilized for that particular function in that particular cell. And then you have proteomics, which refers to the total number of proteins in the cell or in the tissue which you are going to analyze. And then you will not only have the amount of proteins, but you'll have the protein mapping network between the proteins, the production, modification of proteins, the four-dimensional structures, etc. And finally, you have metabolomics. So metabolomics refers to the total number of metabolites in the cell. So any genetic activity or transcription, translation, all of this results in multiple metabolic activity in the cell. You can measure the metabolic activity, and that metabolic activity itself will tell you what is actually happening in the cell and how the cell is taking it from the point of genetics right to the point of action. So all these things are important when you're trying to discuss. And if you look at omics, omics is going to produce what is known as personalized medicine and transplant. So not that personalized medicine is not already there. In fact, I would say that transplant is probably the most personalized type of approach that we are having in our own, what do you call, in our clinical practice. Because two transplant recipients do not behave the same way, even if their background is the same. We have to adjust for their comorbidities, their immunological barriers. You have to adjust the drug dosing depending on side effects and the amount of drug levels that you see. You have to see whether the patient has done an infection or not. So every transplant patient is a big matching issue for us. So that is going to be a problem when you are going to see the transplant patients as such. And finally, what can happen with omics can actually help us to improve the personalization. You can plan donation, you can plan pre-transplant evaluation, you can identify problems, you can predict what is going to happen to the allograft and to the patient over time. But the question is, how are you going to do this? How is this possible? So let us first take a fictitious patient, which I told you about. This patient is diagnosed to have X-linked Alport syndrome. We are planning for a kidney transplant. He's on peritone dialysis. He's in good health, no history of transfusions. And he has a maternal cousin who's willing to donate. She's 35 years of age. The urine shows a few RBCs. And that's the problem that we are having. She's willing to donate, but there has been no genotyping done in the other family members so far. The problem here is, what is the risk for the donor? So now you know that genetic testing now becomes important. We know the genotype of the patient, but we should do a genetic testing for the risk for the donor also. Now, the problem here, what type of genetic test will you do for the donor? For that, we have to understand genetic tests and what type of genetic tests are available and how to choose between these genetic tests. That's the most important thing which you have to know. For that, we have to start with this person called Frederick Sanger. He's a double Nobel Prize winner. He won a Nobel Prize initially in 1950s for synthesizing insulin. No, not actually synthesizing insulin, for actually diagnosing the sequence of insulin, the protein insulin itself. And second time, he won a Nobel Prize for inventing what is known as Sanger sequencing. So the Sanger sequencing actually refers to a genetic test in which the DNA is actually, you are trying to identify what type of DNA and the sequence of the DNA in a very, very, very important and simple type of step. So the first step in Sanger sequencing is you denature the dsDNA using heat, and then you have, when you denature the DNA using heat, you have the two complementary strands will be separated from each other. And you take one of the complementary strands and make multiple copies of this particular segment of DNA. So you can do it only piece by piece. You cannot do the whole DNA as such. You take one particular small genetic sequence, maybe of 300 bases, 200 bases, and then you make multiple copies. Then you attach a primer. So what do you mean by a primer? A primer is something that will start a polymerase reaction and cause multiple copies of gene to form. Once you add the required ingredients, so you have to add a polymerase and you have to add multiple nucleotides, which can actually help in doing that. So the next step is to add this to 4 polymerase solutions. So you have 4 polymerases. Each will have one, they will be filled with actually DNA nucleotides. So these nucleotides will help this DNA strand grow because of the polymerase reaction. But also, these are also infused with this DDNTPs. Now, what are DDNTPs? These are actually stop codons. So if this particular DDTTP is inserted somewhere in this line, it will cause chain termination. So the DNA synthesis will stop there. And C will stop at some other level. So putting this randomly, when these are inserted into this, you have chain termination at multiple levels. It's not as a chain termination PCR. And so it will have chain terminate multiple levels. So what you will get is you will find different complementary chains at different levels. So you can see this chain is this long, 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 and each has got a different type of TTP as its last termination sequence. So finally, what is done is all these final products are passed through a gel electrophoresis, and they will separate according to the size. So the smallest ones will travel longer and they'll reach the end. And then what happens is that you can see the smallest one will have at the end this particular TTP or something. And you can just read off the genetic sequence just like that based on the last protein. So it's very simple how to read it off at the end. Now, this was done actually using manual techniques, but nowadays it's being automated, so you can do it much faster. But there are problems with Sanger sequencing. Even though it is 100% accurate, you can run only short sequences, variances. About 300 base pairs is what is usually done. You can go up to 500 base pairs or a little bit more. But you must imagine that sequencing the entire genome will take a hell of a lot of time if you're using this technique, because the genome itself contains 3 billion base pairs. So 300 to go, you takes many years. And the whole, this time consuming, it's not only it's very time consuming, even though it's got automated. If you look at the human genome project, this project took about 20 years to complete. All the sequencing was done through the Sanger technique, painstakingly by multiple researchers in multiple institutes, thousands of people working around the globe in order to sequence, and it took 20 years to do it. But the important thing is that once the human genome was declared complete in 2003, even though it was complete, that was only about 90% of genome was sequenced by the time. But all current technology NGS would not exist if this Sanger had not been there and if the map of human genome had not been there. Because NGS, everything uses the reference genome as the reference molecule, the human genome as the reference molecule. And all their reports are based on the reference. So if the reference had not been there using this, we would not have modern techniques. So then let us talk about NGS, because NGS is one of the things that we are going to use in regular parlance all the time. So most of the time we are looking at NGS data and NGS reports. So the basic difference between Sanger sequencing and NGS, or next generation sequencing, is that this next generation is like Sanger on steroids. You are going to do massively parallel sequencing. So instead of doing one particular gene segment as we are doing in Sanger sequencing, in NGS you are doing hundreds of genes, thousands of genes, even millions of genes in parallel. So the same test is being repeated in parallel for multiple genes. Therefore, for the time taken to sequence one gene, you can sequence probably the entire genome. The problem is that this generates a huge amount of data. It is not possible for a human being to read this data. You need a bioinformatics platform to actually read the output and produce a particular report. So what steps do you do in this? So there are basically 4 steps in NGS sequencing. I'm just reducing it to very bare minimum because actually the technique is much more complicated. So if you have multiple DNA strands to take, so this year you have about 6 DNA strands. So one of them is taken and you add 2 adapter molecules to both sides. Now these 2 adapters are different adapters and they are important because they are actually acting as the primers in this, and they are actually matching the adapter or the oligonucleotides you're using for the platform here that you're using. We'll come to that a little bit later. So this is creating a DNA library. So this DNA is an actual adapter and you create a library using this one, all these genes separately. So you create a DNA library. But this library alone is not enough. You need multiple copies and you need only then you can get it. So next step is to create a DNA cluster. So this particular DNA, you create a cluster out of it. So you make multiple copies of this using a polymerase chain reaction or using a DNA polymerase or a DNA polymerase synthase enzymes and all that. So what you do here is that you have a clonal amplification of this particular DNA segment. So you have a platform. Now we can see that on this platform, you have multiple oligonucleotides. Now, as I said, these oligonucleotides match the adapter sequences on this. So when you put this particular DNA which you have taken, now this lines up along with this. And this oligo acts as a primer for sequencing the complementary strand here. So you can see that the complementary strand is coming up. And this complementary strand, the other end attaches to the particular oligo adapter here. And so you can see this is what happens. The original strand is then washed off. So what you're left with the DNA complementary strand which attaches to this particular platform. And then you can create multiple copies of this using a polymerase reaction by itself or using a PCR technique. And finally, what happens is that, so once you have done the clonal amplification, created a DNA cluster, you have a final step in readout. So this is readout using different, each particular platform has got different way of reading out. All we need to know that once you have this DNA sequence can be read out very fast on the reader. And this is just one particular DNA sequence. You know that at the same time, you're doing 10 or 100 or 1,000 or 1 million genes in parallel. So all the readouts are occurring at the same time. So you can get a final readout in a short period of time. So here, what we are testing for are SNVs and CNVs. So SNVs are single nucleotide variants. This refers to mutations in single gene, just one gene. Say if you have CTAG, one T is mutated into a C, or it's missing altogether, or it's been replaced by a G, or it acts as a frameshift mutation. There are different types of mutations can occur, which we need not go into right now. The second thing is a copy number variant. This refers to deletions and duplications. So you have, for instance, if you have a particular deletion, then that is referred to as copy number. So because it means either the number of copies of the gene are less or more. And that less means a deletion, more means a duplication. That's why it's called a copy number variant. And other thing is single nucleotide variant. So you have both SNVs and CNVs. So NGS and Sanger can detect SNVs, but they are not very sensitive for CNVs, especially large ones. They can miss it completely. So for that, we need a different type of test for deletions and duplications. So what type of test will you do for that? So you have two types of tests for this. So these are actually the CMAs and the MLPAs. Just a second. Yeah. So these are two different complementary tests which can be used in this patients. One is chromosomal microarray is used for large deletions across the genome. So it is probably the first test of choice for congenital defects in the baby. So you can do, because most of them are due to deletions and duplications and all that, like trisomies and Edwards syndrome and all these syndromic things. CNVs, the CMA is the best test because it can detect the deletion right across the chromosome. All the deletions across the chromosome can be detected. MLPA is otherwise known as a multiplex ligand-dependent probe amplification assay. So that detects the copy number variants at specific gene sites. So if you have a, say, for a disease like ADPKD, you know the gene which is involved in ADPKD. And so you can just do the gene duplication. So these are for smaller duplication and deletions which are missed by the NGS. You can do an MLPA. So in many of these labs, what happens now is that you can do the test. You can do, say, for instance, if you have a point mutation, you say a disease like Alport syndrome, mostly it's your point mutation, you do an NGS panel that is good. But for a disease like HUS or ADPKD, where you have not only point mutations, but you can also have deletions and duplications, you have to do both an NGS and an MLPA as a follow-up test. Don't do an MLPA first because MLPA is not good at detecting point mutations and will miss most of the genes. So you have to do an NGS panel first and then do an MLPA. And many of the labs now are doing a combo test. So if you ask for testing for HUS, they will do both NGS and MLPA and produce the result finally. So that way, so now you know what you do for SNVs, what do you do for CNVs. When you suspect that a disease, you know which disease have got SNVs and which disease have got both SNVs and CNVs. So you can order the tests appropriately. And the genetic lab helps you by doing combo tests and others. Having done, what do we do in this particular patient? So this is a 25-year-old man with a maternal cousin willing to donate. So what did we do for that? So this is the original report in the genotype in the patient. So you can see it's a called for A5 mutation, which means an X link. It's belong to the X chromosome, hemizygous zygosity. And you can see the variant here. It is C stands for the codon, the gene codon, and that is the precise location of the codon, and means that there's a del C. Del C means a deletion of C or cytosine. And this P refers to the protein. It's actually the proline at this level which is being transferred. The rest of it is rather difficult to understand, but you must imagine it's a protein mutation which is also mentioned here. The C and the P both are mentioned here. I know the precise mutation is now identified. So you can do two things in this donor. One, you can look for that precise mutation, but it is possible that this patient has got a mutation in a different gene also because she's a maternal thing, and this may occur in families, especially if there is a consanguinity, which we are not mentioned about in this patient. But actually, the clinical evaluation showed that she had mild vaginitis with trichomonas. And once this was cleared, the hematuria disappeared. But still, she underwent a gene study using a clinical exome panel. That is actually an NGS study. And this was found to be negative. Other genetic tests were not done, and she was accepted for transplant, and surgery was done with an uneven full coastal discharge. The donor is doing very well, even after 1 year of follow-up. So that is the present situation of donor. So now we know about the importance of genomics in the evaluation of donor. You can use genomics also for several other reasons from the donor and the recipient. One, as we have said here, is the selection of donor for kidney transplant. You can use it to identify the cause of CKD. You can predict the risk of recurrence. You can tailor immunosuppression based on pharmacogenomics. These are some of the uses of genomics in transplant evaluation for the recipient and the donor. So following up this patient, 3 months later, he had a baseline creatinine of 0.9. On routine follow-up, the creatinine went up to 1.1 and then 1.3. And then you did a test known as the donor-derived cell-free DNA. And this has found to be 0.13%. The DSA was also done for this patient, was found to be negative. But a renal biopsy was also done despite all this and did not show any features of rejection. Now we look at what is this DDCF DNA test. So this is one of our own studies which we did in a group of 24 patients with multiple causes of rejection. You can see that the one patient who had chronic active ABMR, the value was very high, 2.1%. All the others with other types of injury as well as no injury at all, they had a value less than 0.3. So the DDCF DNA in general, this was the recent Admiral study which studied that, and we found that it for discrimination of rejection, it has an ROC curve of AUC of around 0.8 almost. So it's reasonably good discrimination for rejection as compared to creatinine, which does not distinguish between anything. But the problem is that this actually showed a much better discrimination when it was negative. So a negative predictive value is nearly 90% for this. So probably if the DDCF DNA is negative, it means that there is no rejection. If it's positive, it may be that there is a rejection, but you have to do more studies in order to confirm that. So this is a test which measures the genomic presence of donor genes in the blood, which are donor genes are obviously derived from the allograft. There's no other way in which donor genes can be present. So they just measure the entire genomic pool and find out what is the percentage of donor genes. So more donor genes, there must be some damage to the allograft, which is causing an increased number. And the most important damage is the allograft ABMR, in which this level rises much higher. So you can see it was very helpful in this case to rule out. So we know that now genomics has a role in the diagnosis of rejection in the patient. Now this is one of the examples. Probably we will have much more examples. There were a lot of other parameters which are being studied in these patients and probably which may be useful in diagnosing rejection. So we know that genomics is useful for not only for assessing which donor can be used for the patient, we are assessing several aspects of the transplant recipient, and also in follow-up diagnose rejection in these patients. So what happened to this patient after that? 9 months later, he skipped drugs for a few days as he had to travel. His creatinine moves up to 2 milligram. Very common situation in our patients. Renal biopsy, unfortunately, was not that helpful. It showed a PTC of 1, an IFTA of 30%. And you did an EM also, which showed a layering of the PTC, but only 4 layers were identified. It did not have more than 6 layers to definitely make a diagnosis of chronic ABMR. So we could not make a definitive diagnosis of rejection. So this patient was, you can see this is one of the pictures. This is actually from the BAN classification data. And you can see this is an example of C4D, of actually, no, not C4D. It's an example of PTC1, in which you can see there are only some 3 to 4 cells in each segment. So I need at least 5 to 10 to make a diagnosis of PTC2, which would have made our diagnosis of rejection much stronger here. So what was done in this patient was this test known as a molecular microscope. So this actually is a transcriptomic assay, which sees the mRNA pattern in the graft biopsy. And you can see that this is the yellow triangle refers to the recipient sample. And you can see it's placed here, R4 and R5. And this R4 refers to early stage ABMR. So it is very close. And this is accounts to 2 reference sets, and both of them classified it as R4. So it's supposed to be a highly reproducible test when you're using this in clinical practice. So it will, even if you get medullary tissue, it will still show the same pattern. So there have been a large number of validation studies with this, and they have all shown similar effects. But still, you need more data in order to really put it in the world. In the US and Canada, it is now being utilized in transplant centers, but I don't think it has reached India as far as today. So this is the role of transcriptomics in allograft pathology. So you're going from genomics to transcriptomics. The transcriptomics refers to the entire mRNA upregulated as well as those which are suppressed, and those are measured during gene transcription. And the technique we have shown you is known as MMDX, or molecular microscope, developed by the Edmonton group. And MMDX uses what is known as archetypal analysis. So what you're doing in archetypal analysis, you are taking the extreme values. So supposing you have a biopsy showing one particular extreme value, say in the patients with ABMR, you take it, all of them ranking one particular extreme values. You take that value as the standard and mark these less extreme values around it, and then you have a profile of ABMR. You may have another extreme value for a patient with acute cellular rejection, and you put all the other values which are around it as a analysis of the acute cellular rejection. And then you do a validation of this particular markers on another series of biopsies, and you see whether it works very well in those specimens also. All this is done by bioinformatics platforms, and you can see that finally you get pure data showing that this particular type of presentation means it is ABMR, this type of presentation means it is ACR, etc. And this means that there is no rejection, and so on and so forth. And finally, BAN 2017 has actually said that increased expression of gene transcripts or classifiers is strongly associated with ABMR if thoroughly validated. That's a very important thing. It has validated in multiple studies. Only then we can say it is. But this actually is equivalent to doing a C4D in the assay. That is the benefit of BAN 2017 classification. So this is probably likely to change the interpretation of graft biopsy in the near future. So what happened based on this? So that the patient received 10 sessions of Plex, low dose IVIG, AC pulse steroids, his creatinine improved to 1.5 milligram. Over the next 6 months, it began to worsen. So late ABMR generally doesn't respond to therapy, even though it may initially show, because of the initial response may just be due to the improvement in the ATN and other features. So what happened after 6 months is that he developed fever with cough. A chest X-ray showed a few nodular shadows. He developed abscess over the skin. The culture grew gram-positive bacillary and cocobacilli organisms, some of which were branched. But you can see that the final report is going to obtain after 3 more days. The patient is quite sick. You cannot wait 3 more days to start a specific treatment for this patient. So what should you do? Then it took a different type of proteomic analysis in the data. Proteomic analysis, multiple types are now available for multiple reasons. So you have them in treatment in finding the allograft quality. So before you actually transport the graft and implant it into the patient, you can do a pre-implantation biopsy allograft quality proteomic analysis, or you can do the perfusate. What you use in the perfusate itself, you can take and see what all proteins are there in it. You can analyze it and look at the allograft quality. Then you can look at ischemia reperfusion injury. There is a tissue biopsy proteomic analysis. For DGF, for rejection, for chronic allograft dysfunction, for BKV, for all these things, there are so many studies which have been done using biomarkers, proteomics, exosomes, extracellular vesicles, and all that. But proteomics has become very important in this, and proteomics data is now becoming available. Unfortunately, none of these have actually reached the stage of clinical utilization. But there are several which are showing a lot of promise. It needs more and more validation. And probably in a period of another 5 to 10 years, we will be seeing proteomic analysis in the management of the transplant. But in this particular case, we use a type of proteomic analysis known as MALDI-TOF. So what is MALDI-TOF? MALDI-TOF stands for Matrix-Assisted Laser Desorption, Ionization, and Time of Flight Assay. This is a very interesting type of assay. So you're using a particular matrix in which you are putting this analyte. The analyte is actually, you're taking the part of the culture which you have grown and putting those culture particles into this matrix. And next step is you put a laser into the laser beam to that, and that will cause desorption. So this is actually removed from that along with the matrix, part of the matrix along with it. And then you pass it through an electric field, which will actually destroy this matrix and actually ionize this particular analyte that you have received. So it becomes a positive charge. The proton is transferred, it becomes a positive charge. And next you put it through a mass spectrometer and you analyze it. So the mass spectrometer does what is known as a time of flight analysis. So what it does is that each particular particle is transported across an electric field. So you know it goes from a negative from a positive to a negative. And for each particular particle, the time in which it takes to fly from the positive to negative is measured. And this is charted. So you have multiple molecules from the culture. You don't get just one particle only, you get some hundreds of particles. And each particle will have a particular time of flight. And then you map it out like this. So you see, you get a map. And this is for a Burkholderia pseudomallei. You're getting this. So you recognize this thing. The computer will immediately say this is Burkholderia pseudomallei. And if you recognize this particular is Burkholderia. So every single organism has been tried on this. And there is a pattern associated with it. So if it is a already present molecule, particular organism which you know, MALDI-TOF will find it, and MALDI-TOF will find it in a matter of few hours. So that's all it takes in order to get a particular assay which is there. So that's how this proteomic analysis, which works which is actually present now, which can help you to diagnose infection in the transplant recipient. And the TAT is about 10 hours, the turnaround time. This organism was shown to be Naquadia asteroids, and the treatment was started with meropenem and Bactrim DS. The patient improved over time. And so you know that proteomics, at least in infection manner transplant, is very important now. In the actual manner transplant, probably it may be important in the near future, not in the far future. Everything is happening in real time as of now. Finally, about metabolomics. Metabolomics is the measurement of global small molecules in the organ of interest. Interest in metabolomics has started even after the interest in proteomics. So there are many proof of principle studies as of now, but none of them are as yet ready for prime time. But one example of metabolomics, which are already using the perfusate solution that we have. If you have UW solution, the custodial solution, the Eurocolon solution, all of them have different types of metabolites in that, which are necessary for the allograft to remain active till the time of the transplant. So that's a very primitive use of metabolomics, but that is the only use of metabolomics right now we have in the graft kidney. But I am sure that the requirement for metabolomics is going to grow both in diagnostics as well as in treatment of the transplant recipient. Finally, a little bit about the role of gene editing. So gene editing we have seen in the porcine kidney recently, but what is gene editing and what is going to happen to gene editing? So CRISPR is actually refers to clustered, regularly interspaced, short palindromic repeats. So CRISPR is actually a very interesting discovery. In the 1970s and 1980s, a few Spanish researchers found bacteria which were having some extra genes, and they were actually having regularly interspaced domains, which are palindromic repeats. So they said, what is this? They didn't realize. Finally, they thought these are junk genes and left it aside. But after some time, some people looked into it, and they found actually that the number of repeats increased as the number of infections of the bacteria were there. So they realized a type of an adaptive immunity in bacteria. What happened to the bacteria when they were infected by viruses? The bacteria would finally take, if the bacteria survived the virus, it would take that gene called Cas9 and snip it off at one place and take some of the genes and put it in this memory in the gene pool. So next time what happens is that if the same virus infects, this Cas9 will come about and find this particular same CRISPR type of domain in the virus and cut it at that level. And the virus will be just cut into bits and pieces, and it cannot proliferate inside this. So it's a primitive type of adaptive immunity seen in the bacteria. But what happened is that this was seen to be very interesting, but Jennifer Doudna and Emmanuel Charpentier realized that this could be used for gene editing in the human being and in animals, and for creating newer copies of genes which are without abnormalities, without mutations. And therefore, they won the Nobel Prize for this recently, for these two researchers. And CRISPR-Cas9 has come into gene editing software. And finally, in the xenotransplant, we have already seen 10 porcine kidney transplants. Both of them underwent editing of the genome using this CRISPR-Cas9. And it's so that once the gene is edited at that level, it will either repair itself automatically, or you can put you can replace one thing with another using another software. And once this germline editing is done, this gene remains like that, and subsequent copies of the gene are actually the normal gene, not the abnormal gene. So you don't have to, you do it only once. That's the advantage of doing a gene editing. So more gene edits are possible if required, and therefore we are sure that gene therapy will have a bright future in transplant. So in conclusion, what has happened is we have gone a long journey. We have seen the evolution of omics technologies from Sanger sequencing to NGS, CMA, MLP, etc. We have traced how different omics technologies affect the evaluation, planning of transplant, the post-transplant evaluation, as well as the management in real time today, and what can happen in the future. And we have also seen some speculation on the future of omics technologies, but let me give a little bit more into that. What we can have an omics future in transplant. So imagine this particular lady who is awaiting a transplant. She's on dialysis. So now, right now, what you can do is find a donor, do a transplant, do the immunological workup, etc., etc. But here, what you can do now is, in future, we have a genomic testing. In this genomic testing, you can identify the gene defects in this patient. You can check for a related donor. You also can find other genes which predispose to cardiovascular disease, cancer, diabetes, and multiple disorders in this lady in course of time. You can also do a pharmacogenomics and see what drugs she's going to tolerate and what drugs she's not going to tolerate. Everything can be done. Probably you can do gene editing later on if necessary. So you can do gene edits in allograft, especially if it's a porcine allograft, which is going to be available in future. Already there are companies who are manufacturing porcine kidneys, and this is going to be one of the futures of transplant. You can do gene edits from the recipient, but then this requires multiple ethical clearances. You have to do primum non necessary. They should not be producing any problem to the patient in future. These are very, very classified. There is a non-targeted effects of gene editing. So you may do an editing one, but the same gene, similar type of gene may be present somewhere else. You may edit and create problems. So that is one of the problems in gene editing, which we have to be surmounted before we can do all this. So this is not ready as of now. It's going to take time. But gene editing, the allograft is already ready. It is already present. Then post-transplant follow-up, you don't need to do creatinine. You have urinary proteomics, which are going to come up in future. You can test all that. You can do graft transcriptomics when you're doing a biopsy. Apart from the pathology, you get the transcriptomics. You can do metabolomics and proteomics and follow-up. So finally, as a result of all this, you can have one kidney for life. And that is the final holy grail of transplant. You just do one transplant, and that transplant lasts for life, which is much possible in a very glowing and very important sunrise future of genomics and omics technologies. Thank you very much for the patient hearing, and I'd be happy to welcome questions.
Thank you, sir. Yeah. Thank you, sir. Am I audible? Yeah.
Yes, you're audible.
So
Yes.
you've explained the crux of omics in a very structured manner. In fact, it hits differently when a nephrologist explains it to us as compared to a person who is in the lab and who's a genetic expert. If we will take questions at the end of panel discussion. We'll now move on to the next segment of the panel discussion, because we have a very stipulated time of 1 hour only. We'll now begin with the panel discussion, sir. We are now in the era of precision medicine, which is more demanding for the healthcare provider as well to give accurate treatment for patients. And one such facet, as very beautifully explained by Balan sir, is precision medicine by omics technology in transplantation. For this, we have two very esteemed panelists. Dr. Narayan Prasad sir, who needs no introduction. Sir is a professor and head of nephrology at SGPGI. But and apart from this, I know personally that he is very involved in molecular studies. So he's going to be a very great asset for us for this discussion. Our second panelist is Dr. Manish Balwani sir. He is the Secretary of Indian Society of Transplantation, and he shall be very valuable in sharing his experience as to where are we in terms of omics technology in transplantation. So at the outset, let's begin with our panel discussion with Narayan Prasad sir. Our questions, I'll just display on the screen. Sir, welcome to Nephrocon's panel discussion.
Yeah, thank you. Thank you, Prasad.
Yes, sir. So, we've understood the basics of omics, but moving on to its use in transplantation. So do you believe we are doing genetic studies for recipients very often? These days, we no longer label a patient with chronic glomerulonephritis or chronic tubular interstitial disease. It's more precise. The diagnosis is more accurate. And now that we are moving into precision medicine, do you think that genetic studies should be conducted for living related donors as well? And how soon do you think that this will become a criterion in the continually evolving transplant protocols?
Yeah, thank you, Prabha. This is a very excellent and pertinent questions, and probably this might be evolving in every nephrologist mind. So whether it should be done in all donors. But I think at present it is too premature to say for all. But definitely there are some conditions where we must look into, like selecting the donor from the ADPKD family and any polycystic kidney disease family where suppose the patient donor is relatively younger, 39, 40 years, and by the time the patient did not have any cyst, but whether there is really a genetic predisposition to develop polycystic kidney disease in future. So that kind of the situation, it is very helpful. No question, no doubt about that. But some of the conditions, like obviously you to talk about this genetic predisposition for FSGS kind of, then a Paul gene was very much talked in the literature, but that is not true for our scenario. There are two studies well conducted, one at PGI Chandigarh, another at Madras Medical College, and there are not any single Paul gene polymorphism has been detected. So it is looking like it is very rare in our part of. And then definitely comes at one of our own experience. I am telling you, the patients are coming for the nephrotic syndrome, and in series of the 65 patients of the nephrotic syndromes, we have POL4A3, POL4A5, POL4A4 mutations in at least 35% of the patients. So these are the some genetic mutations we do not know. Some are coming like the pathogenic, some are coming at the variable significance we do not understand. But it is possible that these patients might develop Frank-Alport syndrome in the future. So that kind of things is there basically happening in theoretically when you go into the depth. This is our own data that I am coming, and we have analyzed and just sent for the publication. So that might come in the future. But there are many like autosomal dominant tubular interstitial, the Euromodelin gene, some mutations, that kind of things. And you can see these some of them manifest subsequently in later part of the life. So it becomes important to choose in which group of the patient donors, selected donors you want to choose. And definitely that we like to do in some of the patients, but definitely making it universal is a little problem, particularly in India, except some parts of India where deceased donor transplant program is very robust. Majority of our Indians are dependent on the living donor transplantation, and there are limited number of the donors also, and then getting this exposing every donors for the genetic testing would be problematic. But many of the genes might come of non-significance. Then definitely, if something comes of the variable significance, we must go for the Sanger sequencing, as Dr. Balan has told us. Sanger sequencing one that gives you confirmation about that gene's mutation. So that becomes important. So I don't think at present we should go for genetic testing in every living donors, but in selected donors it is yes, particularly in the PKD family, where you think of some kind of the disease, whether which is running in the family, like autosomal dominant tubulointerstitial kidney diseases. It is possible. If the family history of the kidney disease is there, definitely we should go for. So we should have certain criteria defined in your protocol where we should go for.
Sir, one case we have in our current setup right now, wherein we were advised, the genetic expert, she counseled the patient who had atypical HUS. He was found to have CFHR 1 to 5 deletions on MLPA. As far as I remember, 1, 2, 3. And she had advised that the donor also should go genetic testing. But the donor is around 65 years old lady who has not had any clinical illness so far, any kidney disease. So what do you think, like in this for genetic testing in such donors, do you think it's going to be any valuable by doing her test? Because do you think
these are some of the dilemma of the present testing. When we go for genome sequencing, sometimes this kind of situation comes into picture. Definitely that kind of things needs to be confirmed on the different samples. It is usually advocated that you should get the different samples, go for the Sanger sequencing and see really this is there or not. So only believing on the whole genome sequencing does not fulfill your requirement. So that is one important thing. There we should go for the direct Sanger sequencing and get that targeted gene, whether this is there or not. As far as age is concerned, I can definitely say you one of the patient, 55 years old lady who had hysterectomy, she came to us in anemic stage. We have got biopsy. It was totally confirmatory of thrombotic macroangiopathy kind of the picture. Clinical picture was also fitting into that. We have got genetic testing done that confirmed the CFHR5 mutations. So it is possible that any point of the age, if there is genetic predisposition, any trigger can trigger this. So it is quite possible that even the elderly patient who have genetic, but definitely when you have this variable significance, no significance, you are thinking of this is the age where it is less likely, we must confirm it with the other methods, like the Sanger sequencing, rather than believing on the whole genome sequencing. That is my suggestion. I think Satish Balan will agree with.
Yes. I think, like you said, I don't think every donor will need genetic studies, but definitely these type of donors do genetic studies. And the other problem is what you said. No? Once you have genetic study, what do you do with it? That also requires, that will be a problem if you're doing genetic studies for everyone. You'll have more problems than solutions.
Right. So the next question is, do you foresee urinary transcriptomes and chemokines being a standard practice for diagnosing rejection in kidney transplant? And if that happens, what challenges or issues do you anticipate in implementing these techniques?
So this is a very vast topic, and something has some part of this has been covered by Dr. Balan. So I'm just giving you one example.
Yes, sir.
In transcriptomes, which was most studied, was the Suthantri cells, particularly urinary transcriptomes, purporting enzymes, faciligands. This has been studied well. And that was studied initially as a biomarker, urinary biomarkers, as a diagnosis of the cell-mediated rejection. So subsequently, it is possible that you might have the many biomarkers therein. You can have at least, I think, more than 150 different kind of the transcriptomes and chemokines have been studied. Which was most studied was the perforating enzymes, faciligands. But what happens subsequently? I'm just giving you one example. That urinary things was not replicable in other centers. Even in our lab, Rajesh Yadav is also here with me. We have tried to get this from the urine, but failed to get this transcriptome. So replicability was very much questionable. Many times we have tried. I have contacted the original investigator also, but I don't say it is not, but it may be difficult to replicate. The other things, what happens, like this purporting enzymes and other things. When you have the severe antibody-mediated rejection, you have activations of the NK cells also. And the NK cells can have the same kind of the purporting enzymes kind of the cells. So there you are too much confused. So whether you have to treat as a T cell mediated rejection or it is a severe kind of the B cell rejection where NK cells came into the picture. So there are a lot of overlapping things are there in this transcriptomes. So I think definitely it would be if someone standardized in their own lab, in their own practice, it might be very useful tools to avoid biopsy. But you think our mind or a nephrologist mind or any transplant physician's mind is always very much believable on biopsy rather than going for your. But again, you might have the urinary transcriptomes, you might have some transcriptomes in the blood also, and you might have many transcriptomes in the tissue per se after the biopsy. And I think that after the biopsy, getting these transcriptomes, sometimes it is very confusing to see whether this is T cell mediated rejection, whether it is B cell rejection, overlapping both kind of things. So there it can help out whether there is a mixture of the both of. So urinary transcriptome, it is fine, it will be non-invasive, but I think urinary blood and tissue transcriptomes all might be useful, but I think it is at present for the experimental and research purposes rather than you can go and believe on for the clinical practice.
Right. So the last question for you would be, considering the common use of CAR T cell therapy, a lot of oncologists have started using it and they are seeing good results. Do you anticipate transplant physician in India also adopting it soon for desensitization?
Actually, about 2-3 years before, I have studied these CAR T cells in very depth, and I have taken one lecture also. Onco-nephrology lecture was there in. So what I anticipate, it cannot be used right now for the desensitization purpose. Rather, it activates the T cells activity and will cause more injury. Basically, it is used in oncology practice to as an anti-tumor cells therapy. And a chimeric antigen receptors is tagged on this, then you can proliferate the T cells after getting this, and you can reinfuse and get the tumor cells which is expressing this receptors that would be killed. So rather it activates, so it is otherwise.
Right.
So there are studies in heart transplantation, heart kidney transplantation. There was one series I have gone through during that period of that time when they have tried to treat with non-Hodgkin's lymphoma, B cell particularly non-Hodgkin's lymphoma. They have tried to use, and all 3 patients out of 4, probably 3 had very severe kind of rejection, and sometimes after that the graft was lost. So I don't see at present can be used for the desensitization purpose, but definitely it is used for the difficult patients, for those particularly post-transplant lymphoproliferative disorders or some non-Hodgkin's kind of the lymphoma, which happens in renal transplant recipients, then can be used with all precautions, whether this is going to increase the risk of rejection.
Right.
Because the T cells, when activated, that T cells is used to kill the tumor cells. So when there will be activation of the T cells, that might aggravate your T cell mediated rejection and may cause graft injury.
Right. Right. Actually, this CAR T cell therapy was recently discussed at the ASN also. But again, like you said, sir, they're probably still in the experimental stages. Maybe they
I
will.
have gone through
Right.
one presentation. There was one presentation in some of the conference. The fellow has shown treatment in very severe kind of the rejection. They have some manufacturing unit of the CAR T cells in their own unit.
Right.
So there they have shown their effect of the CAR T cells, specifically designed for the treatment of the rejection. So that I have seen, but that never came into the literature.
Right. Right.
So that's why I was hoping that will come into the literature and then we'll see. But unfortunately, I have never seen that in the literature.
Right. Right. So before we move on to the next panelist, do you have any message to young nephrologists who are, what do you think the future is in terms of omics and transplantation? How soon should we start getting the grip on omics and start practicing it? Hello, sir. I think. Hello.
Manish,
Yes,
Yes,
sir.
sir. Yeah.
this is
Yes.
this question for you.
Okay. Thank you, sir. So I think right now what we are practicing in India, we are doing pharmacogenomics. Like Narayan sir has extensive experience in CYP polymorphism study, has gone through sir's paper and presentation in various national conferences. So where he has clearly pointed out that in homozygous state, we need to reduce the dose of tacrolimus, and in heterozygous state also we need to somewhat reduce it as compared to normal wild expression. So in our practice, yes, we are routinely using this pharmacogenomics when it comes to CYP3A5 analysis, polymorphism analysis for tacrolimus dosing. But I tried to search for this cyclosporin metabolism CY3 4A5, so 4A4, it is yet not available in India as a routine test to predict the cyclosporin dosing. And in India, I think, sir, we are using the genetic test now more frequently. So yes, for younger generation, I think we are gradually shifting towards the omics in transplantation. We are more familiar with the pharmacogenomics and genetics testing in the transplantation.
Right, sir. So with that, I'll ask my first question to you, sir.
Yeah, yeah.
At present, it is very much similar to the previous one. What is the
Yeah.
current status, sir? Like, where are we in terms of routine clinical practice? Do you think it is still in the experimental stages and not many of us are practicing omics in transplantation, or unknowingly we have started practicing and we need to master it in clinical
Yeah,
practice?
see, omics is a very broad term. So as we just Balan sir was telling us, so it starts from the epigenomics to genomics to transcriptomics, then proteomics and metabolomics. So of those all five, the major parts of omics, we are practicing right now in India, I think this pharmacogenomics and genetic studies. But when it comes to transcriptomics, mRNA analysis, or proteomics, protein analysis, or metabolite analysis, we are still far-fetched. So it is yet to arrive in India. We need to touch that. We think so that when it is practically possible, economically feasible, we may be trying to do that. So efforts are on. I think we need to understand till then. We should be very clear in. We should have more clarity in our mind when it is more essential. At what time we need to use it. What are the reasons? What are the specific clinical indications like early identification of acute rejection or chronic rejection, identifying the tolerance. All these things. I think in the future we will be looking the more of the role of its omics in transplantation.
Sir, how are you experiencing a shift in clinical outcomes when we have now
Yes.
started using genetic sequencing pre-transplant for evaluation of recipients, or even
Yeah, so
metabolomics, like you mentioned, TAC? We were
pharmacogenomics. So right now we are doing is for each and every patient this pharmacogenomics CYP polymorphism analysis. And
yes.
interestingly, we have found that 60% to 70% of our population in Vidarbha region have some heterozygous to homozygous mutations in CYP polymorphism. So we start with the one third to one fourth dose of tacrolimus, that is 0.03 to 0.04 for homozygous patients. And for heterozygous, we need around 0.06 to 0.07 to achieve the therapeutic drug level. So we are now experiencing less patients, seeing less patients with the TAC toxicity, and are achieving the drug level with the therapeutic drug level with the just what the doses just I mentioned. So we are routinely doing this CYP analysis before pre-transplant. We have included in our workup. And also, whenever a young patient comes to us with the unknown kidney disease, we usually do the complement level. And if those are low, then we go ahead with the genetic testing to look for the thrombotic macroangiopathy. So till now, we have transplanted around 29 patients who had the genetic mutations for the complement pathway. In MLPA, we see the CFH R1, R3, R5 as the common genetic mutations in the patients who have the end-stage renal disease, but we cannot say whether this genetic predisposition which we found was only due to the TMA, underlying disease was TMA, because this kind of genetic abnormality, be it CFH R1, R3, R5, or complement-related dysregulation genes like in C5, factor C5 related genetic mutations, this can be seen also in IgA nephropathy. So there are the alternative pathway which is responsible. It may be the baseline disease, may be the IgA, it may be the TMA umbrella, can be a typical HUS. So it makes us more aware how should we proceed with the transplantation, whether we should proceed or not. And regarding your question about the donor, so we have transplanted 7 patients with the donors were positive for the CFH R1, R3, R5 as well as the patients. So the age of the donors were around 60 to 65, and we have published it in transplantation proceedings also. So all the donors are fine. We have a follow-up of 4 years of those donors. The last follow-up I think was in the last month only, and the donors are still comfortable. So getting an abnormality, just CFH R1, R3, R5 and labeling it as an TMA won't be right. It can be also due to the IgA nephropathy, or sometimes it can be just a common occurrence of this genetic phenomenon in normal population.
Right, right. Sir, the last question, what challenges and opportunities arise in implementing precision medicine approaches in kidney transplant? Given the interpretation also matters, like you just mentioned that just having a abnormal genetic report does not mean anything unless we have to interpret it also. So
Yeah,
what?
along with the clinical situation. Yeah,
Yes.
so this omics is all about data interpretation. I think they collect the data, we analyze it in a bigger way. We have more and more of data, and
Right.
then we see that how the data is shaping in one form to predict a particular form of disease. So
Right.
it is still evolving. I think there are many biomarkers which are being developed. And
Yes.
those biomarkers are being developed like an assay of 13 biomarkers or assay of 19 biomarkers or 7 biomarkers. So this is all about the data analysis, and they say how the particular number of molecules or biomarkers can be clubbed together to predict one particular form of disease or infection or toxicity like that. So it is really challenging, and the artificial intelligence will have a bigger role in.
Right, right. So we don't have any questions on the YouTube live. So I think it is a very too early to discuss omics and transplantation probably in 2024. But I think maybe by the end of say 5 more years, we will be flooded with more discussions on omics. So thank you, Satish Balan sir, Narayan Prasad sir, and Manish Balwani sir to be here on our panel discussion and to be speaker as well. With that, we'll conclude this meeting. If there is any more questions by anybody, we will forward them to you, and then we can expect a response from you in person maybe. So thank you, sir. Thank you so much for being here with us.
Thank you. Thank you.
Thank you. Thank you, Purva. Yeah.
Thank you, sir.
Thank you,
Yes.
sir. Thank you, all of you. Thank you.
Thank you, Dr. Balan, for excellent presentation.
Thank
It
you.
was
Baba.
great. Yeah.
Thank you.
Thank
Bye.
you. Thank you.
Thanks.
Good night. Yes.
Mhm.